Mitosis is important in cell replacement, organism growth, and asexual reproduction. Mitosis produces two daughter cells that are identical to the parent cell.
From one parent cell, 4 daughter cells are produced by sexual reproduction during meiosis. Each of these daughter cells contains half the number of chromosomes as the original parent cell, making them haploid. They are genetically different from the parent cell due to crossing over, providing the basis for genetic variation.
Mitosis is one of the stages of the cell cycle. When interphase begins, the cell begins a period of growth. The growth period is divided into 3 phases, G1, S, and G2. During G1, the size of the cell increases and the number of organelles increases. During the S phase, DNA synthesis and replication occurs. During G2, materials for the next mitotic division are prepared. During interphase, the cell is engaged in metabolic activity and performing preparations for mitosis. Chromosomes are not clearly distinguished in the nucleus, although a dark spot called the nucleolus may be visible. The cell contains a pair of centrioles, which are organizational sites for microtubules. During prophase, chromatin in the nucleus begins to condense and becomes visible as chromosomes. The nucleolus disappears. Centrioles begin moving to opposite ends of the cell, and fibers extend from the centromeres. Some fibers cross the cell to form the mitotic spindle. Prometaphase begins when the nuclear membrane begins to dissolve. In prometaphase, the proteins attach to the centromeres, creating kinetochores. Microtubules attach at the kinetochores, and the chromosomes begin moving. During metaphase, spindle fibers line the chromosomes along the middle of the cell nucleus. This line is referred to as the metaphase plate. This organization helps to ensure that in the next phase, when the chromosomes are separated, each new nucleus will receive one copy of each chromosome. During anaphase, the paired chromosomes separate at the kinetochores and move to opposite sides of the cell. Motion results from a combination of kinetochore movement along the spindle microtubules and through the physical interaction of polar microtubules. During telophase, chromatids arrive at opposite poles of the cell, and new membranes form around the daughter nuclei. The chromosomes disperse and the spindle fibers dissolve, and cytokinesis, or the partitioning of the cell, may also begin during this stage. Cytokinesis results when a fiber ring composed of a protein called actin around the center of the cell contracts, pinching the cell into 2 daughter cells, each with 1 nucleus. The 2 daughter cells produced are genetically identical to the parent cell.
Meiosis is made up of 2 divisions, meiosis I and meiosis II. In meiosis I, homologous chromosomes and synapses become closely associated and crossing over takes place in prophase I. The nuclear membrane disappears in metaphase I, and microtubules form a spindle. These spindle fibers attach to just one side of each centromere, and microtubules from opposite poles attach to the 2 homologous chromosomes. Each of these homologous chromosome pairs line up on the metaphase plate. The chromosomes are pulled toward the poles by the shortening of the spindle fibers in anaphase I. Both sister chromatids move with them. This ends with each pole having a haploid set of chromosomes with 1 member of each homologous pair. Nuclear membranes reform around each daughter nuclei, containing 2 sister chromatids per chromosome attached to a common centromere during telophase I. These 2 sister chromatids are not identical because of crossing over. The second division of meiosis takes place in meiosis II, gamete formation. The nuclear envelope breaks down and a new spindle forms in prophase II. DNA does not replicate. Spindle fibers then bind to either side of the centromeres in metaphase II. The chromosomes align in the center. In anaphase II, the sister chromatids move toward the opposite poles when the spindle fibers contract, dividing centromeres. The nuclear envelope forms again in telophase II, around each set of daughter chromosomes. The resultant is 4 haploid daughter cells.
To summarize, mitosis divides one cell into 2 daughter cells, while meiosis divides one cell into 4 daughter cells. Mitosis conserves the chromosome number, while meiosis reduces it in half from diploid to haploid. Mitosis produces genetically identical daughter cells, while meiosis produces genetically different daughter cells due to crossing over.